Hello, everyone, and welcome to today's live broadcast, Point of Care Genomics, a New Horizon in Personalized Healthcare, presented by Marcel Dinger. He is the founding CEO of Genome.One, head of the Kinghorn Centre for Clinical Genomics at the Garvan Institute of Medical Research and conjoint associate professor at UNSW Australia. I am Marjorie Torres of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom or center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Marcel Digger. I will now turn the presentation over to him.
Hi, everyone, and welcome to this presentation. It's really exciting to be able to give this to the group today because I've got some really interesting new work that I'm going to present and where we're and our thoughts at the moment on point of care genomics and this new horizon in personalized healthcare. Okay, so let's get started. So the human genome, as some of you will be aware, and you may recall from your days back in high school biology, we have 46 chromosomes and about 6 billion base pairs of DNA that are wrapped around amongst these across these different chromosomes in different parts. 23 of those chromosomes that are inherited from your mother and 23 from your father. So all of the information that's in there is really what powers everything around the development of an individual, right from a single-celled individual from when you're conceived all the way through to being a fetus, a child, a baby, and growing up. And it's actually really remarkable to think about all of that information and just how much of it is in there in terms of how it changes throughout your lifetime, and even the subtle things like how your eyesight might deteriorate, or hairlines recede, or even the graying of a beard, as you can see in the image there of the two identical twins there, of just how many of these different characteristics of individuals are actually ultimately programmed inside our DNA. The data is huge. We're talking about, as I said, 6 billion characters of information. If you actually, to kind of put that in context, if you had printed that out in a 9-point font on just normal sized sheets of paper, it would stack up to 300 feet or 100 meters tall. So there's an enormous amount of information in there. Of course, most of this information is about what makes us human and what makes us different to other species and life on Earth. But it's also the differences between the DNA code that's between yourself and somebody else varies by about 4 million or so of those 6 billion characters. So that explains most of the variation between two individuals, and most of this variation is normal. However, a lot of the information that's here can also have an impact on health and disease, and that's the real interest in genomics from a healthcare perspective. So let's go through some of these things. So the human genome, as I mentioned, actually underpins most diseases and influences how we respond to virtually any medical condition or treatment. So I guess what I'm saying there is that we're not just talking about individual hereditary diseases, but even the way that our body responds to disease, like whether that's how our body responds to different kinds of medications, or how our bodies even respond to other medical interventions, such as surgery or infection. So the real value that we see now in the human genome goes beyond just the diagnosis of disease, but also how it can be used to inform clinical care, particularly, of course, in areas such as rare disease and cancer. One of the areas I'm going to focus on today is the expectation and anticipation of the ability to be able to sequence our genome, which has come down so much over the last 10 or 20 years. Now it's possible to capture this data quite easily, but interpreting it and using it in healthcare still remains a huge challenge, and that's part of what I'm going to present today. So worldwide, this is becoming increasingly recognized, this extraordinary impact that human genome sequencing has had. Worldwide, governments have invested billions and billions of dollars of trying to understand what the impacts are of genomics in healthcare. So especially some of the major projects like in England, the Genomics England project, in France, there's a major project. The NIH are running a huge project called the All of Us Initiative, and so on. And of course, including in Australia, where there are several initiatives involved in the application and implementation of genomics in healthcare. So as I said, the price of genome sequencing has come down enormously over the last 10 or 15 years. This is mostly a consequence of next-generation sequencing, massively parallel sequencing, which really started to become available in 2007. Since then, there's been a hyper-exponential drop in the cost of whole genome sequencing, right down to where we are today, which is where the data can be generated, that is, the whole genome can be sequenced for about $1,000. So what makes this test quite so remarkable and distinct from other tests that have come before it is really that it covers many other tests that have previously existed. So this one test, a whole genome sequencing test, potentially replaces, in fact, does replace thousands of existing genetic tests, many of which would not ordinarily even have been available in the healthcare to healthcare practitioners. So in this regard, there is so much more information in terms of a geneticist or a healthcare practitioner doesn't need to know nearly as much as they would have formerly needed to understand to be able to even work out which test to order, which is what how things were done prior to the access to whole genome sequencing. So you can the so-called hypothesis-free diagnosis, where you have all the information in front of you and then can really make the best possible diagnosis and healthcare decision from there. There's also, of course, the ability to reanalyze data. This is very unusual. Most tests, tests are done, but then there's no further opportunity to really make use of that information. But the genome is unique like this, that as we learn more about the genome, we can keep testing it again and again and querying it again and again for new health information. It also, of course, has huge value outside the medical system in a research context. So the other characteristic that makes whole genome sequencing or genomics peculiar or very different to other technologies that have come before it is that it has an impact over so many different areas of life and healthcare, all the way from planning a child in the context of carrier screening, all the way through to understanding what caused the death of an individual, so-called molecular autopsy, and everything in between as well. So understanding like the diagnosing cancer, guiding the treatment of cancer, understanding which diseases we're predisposed to, and diagnosing diseases, which medications we respond differently to, and so on. Okay, so I'm going to talk about some of the key opportunities that are really in front of us today in terms of how genomics can be used in healthcare. So the main one, of course, is the diagnosis of genetic disease. As you might expect, sequencing a whole genome is a very powerful way of diagnosing disease. Cancer, of course, the molecular stratification of cancer, by actually sequencing tumor DNA, we can understand what the drivers are of a particular cancer, and then use that information to actually guide the treatment of cancer. And finally, of course, and this is perhaps one of the more exciting areas that really holds the most promise, is in terms of its impact on healthcare, is actually to prevent disease and to really personalize treatment based on genetic information. All right, so first I'll just speak briefly about genetic disease. So what are the main opportunities in this? Is really in the areas of rare disease. Rare disease, 1 to 2% of individuals are born with a severe genetic disease. Many of these are undiagnosed. Diagnosis can be incredibly powerful for managing the symptoms or managing the conditions of for a patient by having a diagnosis. It can also be very valuable for the reproductive choices for future children from a family. The diagnostic rates have been transformative in this area, with previously many of these diseases remain undiagnosed, but now diagnostic rates are in the area of around 50%. And this is an area that has already really started to have a significant role in healthcare. So what are the challenges in doing this? This is, as I mentioned at the start of the presentation, there are billions and billions of characters of information in our genome. And the real challenge of what we're really trying to work out is which one of these is actually the cause of a disease. It's a real needle in a haystack kind of problem, where you've got millions of variants of things that are just different between individuals. But what we're really interested in is which one is the cause of a disease. And many of us carry actually damaging mutations in our genes in any case, so it can be very difficult to work out what's just normal things that our body is able to compensate for versus those that are actually pathogenic and the real cause of a disease. So at Genome.One, what we were trying to achieve, and this is very similar to other organizations in the genomic testing field, is to try and make this actually accessible to healthcare. Obviously, a healthcare practitioner can't look at thousands and thousands of characters of DNA or to interpret a genome, so it has to be made really easy. So the idea is that is developing really a genetic test that makes it simple to order a test and to return that information back in a way that it's actually meaningful to a healthcare practitioner, and they can use it to guide the management of their patient's condition. So this was a very long process for us, and we were one of the first companies in the world, along with others, that really brought this together and made genome sequencing something that could be done in a safe, responsible approach that fitted in with the existing healthcare system. I won't go through all the details today of all the different steps, but in essence, it's everything from counseling a patient in terms of what they can expect and what the implications are of having a genome sequenced, all the way through to sequencing the genome, analyzing the data, and working out, as I mentioned before, what the pathogenic changes are in the genome, and then ultimately putting this into a report that can be acted on. So we try to do this through a platform that we've developed, where effectively you take not just the genome, but you also use all of the information that's available, the so-called phenotype, to be able to drive the interpretation of the genome, to be able to find the most accurate possible answer. So today, in our setting, we're able to diagnose a huge range of different diseases, from inherited diseases, childhood syndromes, heart and kidney, renal conditions, and immunological disorders, and so on. But as well as that, we've now also started working towards how we can actually predict disease in individuals as well, by looking for genes that significantly increase the susceptibility to disease, such as cardiac disease and cancer, and also those that lead to an adverse response to medications. So I'm just going to give some context to this. I'm going to give an example of a diagnosis that was undertaken at our facility when we launched a couple of years ago. So this was a boy whose name was Alan. We've got permission to use the story, of course. And at the age of 3, he'd been diagnosed with a very rare condition, which was an autoimmune disorder. It was attacking his own blood cells. It was not understood what the cause was of the disease, nor how to treat the condition. At the age of 7, his options were really starting to become very limited for the family, into a program to have his genome sequenced with us. And so it became very urgent because he was in intensive care at the time, and so there was a real need for a diagnosis. We were able to find a diagnosis for a particular gene called LRBA, and this actually connected to coincidentally almost to a drug that had actually been trialed to treat patients with this same condition. And it had promising results. So in this case, the drug did work, and it had a really transformative impact on Alan's condition, and actually really put the condition out of danger to his life, and enabled him to start going back to school again for the first time. So this really shows just how powerful this technology can be, and how it can really change lives. As you can see on the next slide, this is Alan with his father, literally in the back home and playing with lightsabers and basically doing the kinds of things that normal boys do. So to give some further examples of this and how it's been applied in our context, so we've undertaken sequencing of individuals with intellectual disabilities in an area of Liverpool Hospital, which is a large regional hospital just southwest of Sydney. Now, here we again just looked at what was the impact of being able to provide diagnoses for these children. So we sequenced about 26 or so families, and as you can see, we diagnosed about 12 definitively. Some of these which had undergone genetic investigations for years and years, sometimes as long as 17 years in the most extreme case. And we were able to find a diagnosis in just a matter of weeks using genome sequencing technology. So that really just makes a point of just how powerful this can be. Okay, so moving on, I'm now just going to talk briefly about one of the other areas. I won't talk about cancer today, but I'm going to talk about disease prevention and treatment personalization, which is a really exciting domain in genomics at the moment. So Genome.One developed a test, and this was involved with using expertise from genetic pathologists, molecular pathologists, clinical geneticists, and scientific expertise to really try to answer that question: what can we test in an individual's genome that doesn't actually have any particular symptoms for disease? So, and where the information could be returned in a way that it was actually actionable by healthcare practitioners. So this differs from many other kinds of testing that just that are more associated with the disease. This is where we're really looking for mutations or variations in genes that have a high chance that if there is a damaging mutation, that it will cause disease. So in this test, we test for about 169 genes associated with cardiovascular conditions, 56 associated with cancer predisposition, and hundreds that are involved in testing for adverse reactions or unusual responses to medications. All of these conditions that we test for are for treatable conditions, or where the patient or an individual can be managed to reduce the likelihood or incidence of that disease, really with the objective of leading a longer and healthier life by having knowledge of this information about your genome. So here are two examples that give some idea around this. So of people that individuals, their names are changed here, that we've had seen through our service, and gives just an example of the impact that it's had on these individuals' lives. So first example we'll just go through is Linda, who had her genome sequenced and found a variant that could cause a reaction to a general anesthetic. So she hadn't actually had a general anesthetic herself before, but she had a child who needed to have a son who was having a particular procedure, a tonsillectomy, which would require general anesthetic. So she was able to inform the anaesthetist about that, and they were able to accordingly change the type of anesthetic used on her son to avoid any risk of having an adverse reaction to that anesthetic. And in the second example, there is Malcolm, who also had his genome sequenced, and he had very high cholesterol levels, and we were able to identify that he did actually have a genetic variant that was linked with familial hypercholesteremia, which can increase their risk of cardiovascular disease. As a result of this information, and combined with this pharmacogenomics report, we were able to guide, or his healthcare practitioners were able to guide the best possible medication to manage his condition. So those are really nice examples of where you wouldn't have been aware of this information without in the absence of this genetic information, and it's really made a difference to how these patients' health was managed. So when will this start to become more of a routine part of healthcare? So we're seeing all the time, like as I mentioned, we test now for just a few hundred genes, but in the future, it's very likely we'll be able to test for more and more different sorts of conditions and be able to return these in a valuable way back to the healthcare practitioners. So if we look at these curves there, as you can see there on the left, we are seeing a rapid decrease in the cost of sequencing, and in parallel, we're seeing an increase in the value of the clinical value of a genome. And I think that we'll at some point reach where these curves really intersect, and where it will be truly valuable for any individual to have their genome sequenced and to use this information to guide their care. The picture on the right there just gives an idea of just how fast the growth is in our understanding of different genetic conditions. So this just shows the number of genes associated with schizophrenia and how this has changed just over the last 10 or so years, or 8 or 9 years, going from understanding very little of the genetics of this condition to actually now having a good understanding of the genetics of a much better understanding of the genetics of schizophrenia. So one of the main challenges that's really faced that we do face today in using genetic information is really in this spectrum of understanding which genetic variants are what we call monogenic, which means that a single change is the cause of a condition, and what the impact is of environment on this. So some conditions, like say, for example, to take an extreme like cystic fibrosis, a particular change in the CFTR gene, which causes cystic fibrosis, will mean that if that change is there, it will always lead to disease. But there are other genes where single changes don't fully cause or may not cause the disease. And this is a so-called penetrance. So some of the cardiovascular conditions, for example, are good examples of this, where changes in the genome may manifest in disease in some individuals and not in others. And then across this as well, it's not just the environment that has the impact on this, but it can be other genes that individuals carry that can have an impact on how different variations impact on disease. And this is really where a lot of the research is happening today, is to understand more and more about what the individual changes in their genome cause, and how we can better predict how changes to the genome relate to disease. Okay, so the way that we kind of think about this, and to go back to, as I mentioned earlier, is how do we go about this? How do we go from these having these 6 billion characters and of information and working out which one is related to a particular disorder? So what we do is we really are bringing together these 3 threads of information. We can look at the genes themselves and the variants on them, and predict what impact a variant may have on a gene, and in turn what impact that will have on the shape of the protein that it codes. So that's the so-called pathogenicity of a variant. We similarly, we can also look at filtering variants by looking what is actually normal, what exists commonly in a healthy population, and by looking at comparing one individual's genome to the genome of thousands of people, we can actually start to predict and understand the difference between what is normal variation and what's damaging variation. And finally, is the introduction and use of phenotypic information. So that's the actual physical characteristics themselves. And this can be a really important component of being able to accurately interpret the genome. So this is really about asking the right question at the right time to give the best possible answer from a genetic analysis. So this is an area that's been a challenging one, because, of course, sequencing a genome, as I mentioned before, is not necessarily that difficult, and now is becoming an increasingly commoditized in terms of being able to sequence a whole genome. Is the instrumentation is certainly available to do this. There are many laboratories around the world that can sequence genomes, but analyzing the genome and actually extracting the health value from it is a very different story, where the nature of the analyses can have an enormous impact on the overall cost at the end. So if you're spending tens or hundreds of hours of specialist time to interpret a genome, it's not scalable, and of course ultimately makes for a very expensive service. So a compromise needs to be found in terms of how we develop analytic solutions and other automated approaches to being able to still accurately and effectively interpret genomes, but do it in a cost-effective way. So as I mentioned before, one of the ways that we do this is by actually is how we can vary from the millions to the individual ones that have an impact. So to that end, this is something that we did at the Garvan Institute, where I also have a position, as well as the Genome.One, and where we sequenced the genomes of 3,000 healthy individuals. These were individuals that had no history of, they were all over the age of 75. They had no history of cardiac disease or cancer or any other neurological disease. So could be sort of, somewhat defined as being healthy individuals without any strong predisposition to disease, because I guess they've made it up to the age of 75 without being affected by any of these. So we can actually use this now as a reference. So when we sequence a genome of an individual asking a particular question, we can compare it to these thousands of genomes to get a feeling for whether we're picking up variants that are particularly pathogenic or not. So this was a huge computational exercise. We had to call variants from these 3,000 genomes and compare them. And there's some statistics there you can see on the screen just to give an idea of the scale of this. We used the Amazon cloud to do this. And combined, this was really pretty big data, almost a petabyte of data that we put onto the cloud, and we were using thousands and thousands of CPUs all in parallel to make all of the computations to extract the differences from these thousands of genomes out to create this sort of database of information. So this information is available, and there's a platform that we call Vectus, the Vectus cohort platform, that's available at sgc.garvan.org.au. It's freely accessible, and you can actually browse through these thousands of genomes and look at the individual variations across different genes. The other area, as I mentioned, and that's also one that we've invested a lot in, and I think is a really important area for interpreting genomes, and that's being able to understand the phenotype of the individual and their conditions. So this is a particularly challenging area to really make use of, because phenotype, unlike the language, I suppose, that we use for genomes, which is A's, T's, C's, and G's, there is no common language to describe different sorts of conditions. Just to give an example of that, this is if you think about a condition such as, the technical term for this is a skin condition, is palmoplantar hyperkeratosis. In an individual, if they looked at it, it sort of looks like calloused hands, so you might call it like thick skin on your hands. If you the same condition in a mouse, which is obviously where a lot of research is done in medical research is done to understand the impact of different types of variations of genes, it would manifest itself as ulcerated pores. So how do we bring all of these kinds of things together? So a lot of the work that we've done and others is to try and unite this kind of terminology and harmonize it in a way that we can actually use it in machine-led systems, where computers can then effectively read more of this phenotypic information and make use of it to drive the interpretation of the genome. So essentially what our process starts to look like is that we can take now information from electronic medical records, and we can extract common terminology from this and use this to structure this into what we call structured phenotypes. So you may have like a particular term like macrocephaly, which is a technical term for a large head, and someone else might have a term that says big head, and this all becomes harmonized into a single medical term. This can then be related to genes, which can then in turn be used to, target which genes and where to look in the genome. So what we do is that we use information from the medical record to ultimately drive the interpretation of the genome itself. And this provides a more accurate and direct way of being able to analyze genes and return information that's truly relevant to the condition and back to the healthcare practitioner. So to kind of move from there, I just wanted to briefly mention some of the impact around this in both our clinical care in terms of what where the end game, I suppose, kind of is, how this relates to medical research and the industry of the actual testing process. So I guess in previously, pre-genomics, developing a new medical test was a long, complicated process where you would sequence dozens of individuals. You would try to, identify a common genetic cause and then ultimately develop a test, and then that test would be rolled out into clinical care. But what we're really seeing today is that this type of testing is being developed now in a very different way, because we can sequence the whole genome almost in a short space of time. And we have all the information there. We can actually translate new research very, very quickly into the clinic. So a new disease association doesn't necessarily any longer require the development of a new test. The test is already there. It's just a genome sequence, which we can do. It really then just becomes the application of interpreting the application of the information to interpret the genome as quickly as possible. So this really becomes much more about managing information and managing data than it does become about the traditional style of testing. Which brings me to the last part of the talk that I wanted to talk about, which is where we really see this all going. So today, as I said, it's an amazing feat of how far we've come over the last 10 years. It is now possible to have your genome sequenced, but it's still its application, as I mentioned earlier, is really focused very strongly around genetic disease, and it's limited to some extent by access to these very specialist practitioners. So the cartoon at the top there just really shows the sequence of events that has to happen today in order to get a genetic diagnosis. You have a specialist referral, then you see a genetic specialist counselling. You have to collect a blood sample, see that the genome is sequenced, it's interpreted, reported, and then it ends up going back again to the GP and eventually leading to a clinical action. And this whole process still can take, months to go through. So to really have a profound impact on healthcare, what we anticipate the future really holds is where a genome is ultimately sequenced just once in a lifetime. And we certainly do see that the cost of genome sequencing is going to keep coming down, to the point where, if you think of an analogy to other kinds of technologies like photography, which used to be expensive to take a photo, but now it's just like if you think of your smartphone, and a photo is something that effectively costs almost nothing to take. We certainly can see that genome sequencing will go in a similar direction, and at some point it will become almost a trivial amount of cost to sequence the genome. So this data will be available to us, but then it really comes down to how we use it. And the ideal is, of course, that once the information is there, that in any routine visit to a doctor or specialist or any other decision involving your health, that the genome can actually be used in context to be able to guide your care. So, for example, if you're having a medication prescribed, that your genome is being interrogated to see if that medication is going to be metabolized in a normal way by you as an individual, whether a different medication may be more appropriate, and so on. So it's this true personalization of healthcare based on genomic information that where we see that the future is heading, which is just a pretty remarkable thing to start to think about, and how this will ultimately be implemented. So going forwards into the future, I guess that we're seeing more and more, it's not just about genome sequencing, we're also starting to see medical devices having an increasing impact on the health system, whereby these wearable devices, such as different types of biomonitors and things like Fitbits and so on, and these other biomonitoring devices and wearable devices, ingestibles that are all able to take real kind of like almost real-time information about an individual and about your health, and how this is now starting to become more and more intersected and made use of by combining it also with genomic information. So to really truly start to personalize this, and it really starts to transform the way that we think about the way that healthcare, that we interact with the healthcare system. Machine learning and deep data analytics is becoming increasingly a major part of all of this. It's becoming, I guess, more and more clear as well that it's no longer possible for any single practitioner to have all of the knowledge they need with all of this new information that's now become available. And so what we're actually starting to see now is that computers and deep learning are really anticipated to have a much larger role involved in directing and informing healthcare. So where we see the future going really is that all of the data that's out there from individuals will become increasingly both aggregated, and used to really inform clinical decision making. In an optimal world, I guess what we would see is that the information from and outcomes from any patient interaction are actually used to inform the next patient interaction. So, for example, knowing which medications work best for which conditions on a population scale, and actually using that information to then actually start to drive how the health of individuals is managed based both on their genome and all of the information that's available. So to start to sort of wrap that up and to start to think about what the future of healthcare might look like, and so certainly what we foresee is that the genome will start to increasingly inform healthcare, and that this should be able to extend well beyond just rare disease and genetic disease diagnosis, but also to other domains, cancer, cardiology, nephrology, neurology, ophthalmology, orthodontics, and even things, and ultimately general practice and lifestyle, where genomic information can be used to drive all of or inform the decision making in these domains. So in summary, and I'll wrap up with this, is that whole genome sequencing is a powerful technology. It's a single test that can diagnose potentially any heritable disease. It can tailor drug choices, and it can also be used for predictive purposes to predict health outcomes and better manage an individual's health. It's contextual. Reading a genome in and of itself can be useful in some situations, but by far the most effective way of interpreting a genome is actually bringing it into the context of other health information and using other health information to actually direct genome queries, or the way that we interrogate and interpret the genome. There's a huge scientific challenge still ahead of us in decoding the genome. We typically only use maybe 1 to 2% of our whole genome in a genetic interpretation when we interpret the genome. Most of it, basically science still doesn't really understand what it all does. Many of the millions of variants in the genome, we don't actually understand their functions or how they relate to health and disease. There's still a lot to learn going ahead. But given the millions of genomes that are being sequenced around the world, I think there is an acceleration to understanding more and more of how the genome works. So another interesting area, I think, is that clinical genomics has been remarkable in the way that it has drawn together clinical and research domains, and it's really been quite disruptive to both. So we've certainly seen this in our centre, the bringing together of medical research and clinical investigation has been really powerful, and I think it's actually a fundamental component of precision medicine, is being able to bring together both those clinical threads and the latest medical research, and really bringing that together. And that's really where the most power, I think, will come from, in how genomic information is used in the future. Because ultimately, we are actually trying to use information from an individual. So there's always this challenge in bringing together all of the evidence that's needed to drive interpretation, which has traditionally always been based on large clinical trials and studies. But now what we're really doing is we're saying, well, what does this mean for this individual, and what does the variations in this genome mean for them? So I'll leave with that. Remain forever excited about genomics and its potential to transform lives. I'm happy to take any questions. You can contact me on Twitter or by email, or just visit our website. Thank you.
Thank you, Dr. Marcel Dinger, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your questions into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what do you think might be the implications of this sort of testing on a person's health and life insurance?
Thanks. That's a great question. So the implications are different. I mean, obviously in different countries around the world. In Australia, which I can speak to, is that health insurance, which is like a non-risk rated insurance, is community rated, so there's no impact there on health insurance. But on life insurance and other forms of risk rated insurance, that kind of information can be used in the same way that other health information, like family history, is used today. Governments around the world, and some have already put this into place, have actually now made it so that genetic information and predispositions information to disease can't be used to affect premiums in life and other forms of risk-rated insurance. And I think that other countries will follow suit, and that certainly seems to be the trend that's happening around the world.
Thank you for that answer. Our next question is, when do you think genome testing will become mainstream?
So, as I indicated, there's some real challenges around this at the moment, and most of them are associated with workforce. That is, the individuals that are actually available to be able to either interpret a genome or to be able to properly counsel individuals around this. So I think it will be really essential for, I think a key part of this will be software and integration into medical record systems to be able to really both empower physicians, but similarly also individuals and patients to be able to use this information. So there's a long journey ahead, but I think that we are actually starting to see more and more of this happening now. So perhaps over the next 5 or 10 years, I think we will really start to see genomics used more and more routinely in healthcare.
Okay, it looks like we have time for one more question. Our next question is, what is the difference between whole genome sequencing and genetic tests you can order from the internet, like 23andMe?
So whole genome sequencing really actually sequences all of the really the full genome, all 6 billion characters, whereas some of the so-called direct-to-consumer tests, which are just tests that can be ordered over the internet, normally quite cheap as well, maybe for $100 or $200, those are not whole genome tests. They just actually look at a very small percentage, maybe 0.1% of variations in the human genome, and then they correlate or associate these with different kinds of disease. And that information actually comes more from population scale analyses, and they don't actually identify, or at least by and large, they don't actually identify individual damaging mutations to genes. So they can't actually predict with any real accuracy or medical accuracy what genetic conditions an individual is affected by. So they can't be used for diagnosis, basically. And many of them are not supported or can't be used in the context of actually guiding healthcare either.
I would like to once again thank Dr. Marcel Dinger for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Bye-bye.